I was just going to go and have another look at Mauve. Somebody asked me the question, what if you're working with a species that's never been sequenced before? What would you use as a reference sequence? And there are a couple of issues with Mauve. So what I was going to do was just have a look. I do a lot of alignments using bacteria. I didn't ask what organism the person was working with, so I'll just do it from the perspective of bacterial genome. So the goal was to identify a suitable reference sequence and then to reorder the sequence genome according to the reference sequence. One of the other things that was asked was, can you save the alignment so it can be stored in a FASTA file or FASTA file format? So I'll have a look at that. One of the issues was using progressive Mauve, the contigs are aligned in order or numerical order, so 1, 2, 3. So on, and so they're not actually according to the reference, but I'll go through that in a minute. Okay, so what I was thinking was this part here, if you're working with a species that's never been sequenced before, what would you use as reference sequence? So there is one place that you can go to try and find out what organism you might be working with. You go on to rMLST, and if you go on to species ID, what you've got here is ribosomal MLST multi-locus sequence typing. This tab here identifies species, but just on this side here it says rMLST is an approach that index variation of 53 genes encoding bacterial ribosome protein subunits. So this can help with typing and identifying, well, species, if you like. So if you go on identify species here, you don't need an account to use this part. Click on this part here. Now, what I've got is an example which I've just called unknown. So looking at this, it's just a sequence, FASTA file, nucleotide sequence, FASTA file. Put that one in there, and then press submit. So it's just scanning. So it should look through across 53 loci in the genome. So what it's telling me here, it thinks this is Neisseria subflava, 100% confidence in that. Then if you look down this list here, this gives you a bit more information as to how it's reached that conclusion. So these are the different loci here, down in this column. The allele, so the variant of this particular loci, position in the genome and so on. So if you wanted to, you can export that information. I'm not going to. I'm quite confident that this has identified this as Neisseria, in particular subflava. So the next thing I do, if I go onto the NCBI database here, just type in Neisseria subflava and then click on genome and hit search. What you have here is genome assembly and annotation report. So if I tap on that, it'll bring up a list here. If you look down here, you'll see some of these are complete. So these are complete genomes, of which there are 2, but there's also a number which are in contig form. So these are not completely assembled. For a reference sequence, you'd want something that was complete. Hopefully, once you've identified what it is that you're working with, you might be able to find that there's a complete genome for it. So if I just take this top one here, link to genome, click on that. And then at the bottom, just go down, click on GenBank. So this is the complete genome for this particular strain of Neisseria subflava. I think one of the things I found when I was using Mauve, I had a similar problem where I couldn't get it to run, and I didn't know what I was doing wrong. If you go send to file, now you've got this here, it says GenBank. You can create a file in GenBank format. If I just do that quickly, if you open this, what you'll see is there's nothing in there that you'd really be able to use. So that's where I was going wrong. I was downloading what I thought was the GenBank file, but it wasn't the full GenBank file. So it's empty, it's sort of void of useful information for your next stage of analysis. So just going back, the one you want to click on, complete record, file, from the drop down menu, click GenBank 4, create file. And then if I just open this, what you'll see is that this is the full file. So you can see here, this is the header information. Then as you go down, you've got all of the annotations. So the predicted proteins for each of the genes in there. And then right the way at the bottom, you've got the full nucleotide genome sequence as well. So this is the one that you want, the GenBank 4. If I just save that, I'm going to put that onto the desktop in a folder that I've called Mauve. So if I close that now, So you might have already have that or suss that out, because I know sometimes you can get errors cropping up with Mauve. Obviously the file that you put in could be one issue, but there might be other issues. I'm not sure. You might want to try obviously where you save your files to as well, if there's an issue actually Mauve accessing them from a particular pace on your hard drive. You could try keeping them all in one place there on the desktop while you do your analysis, just as a thought. But if you go into Mauve, this is the Mauve main panel. Here, File, Align with Progressive Mauve. Now if you use that one, that'll then just align the contigs to the reference, but it won't reorder them. So what you want is the Mauve Contig Mover tool, which is here. So if you go in Tool, Move Contigs, and it will ask you, choose a location to keep the output files and folders. I'm going to put this onto the desktop where I've prepared the folder, which I've called Mauve. So if I just open that and press OK, and then it will say the reordering will begin when the start button is pressed, and a bit more information. It can take a bit of time. How similar it is, the sequence that you're comparing to your reference will obviously determine how many iterations of reordering will be required. So if it's quite similar, obviously it'll take, well, there's lots of contigs, it could take quite a lot of time. You can only align 2 at a time. The first one that you want to put in is the reference sequence. So just identify where that is on the desktop. There's that one you add in your unknown. In this case it's in FASTA format. Put that one in there and then press start. Okay, so when the reordering is done, you'll get a box coming up saying the reordering process is done, the results are displayed, and data is in an output directory. So if you click OK on that, here you can see above, this is your reference sequence here, and your unknown below. So it's ordered the contigs now quite nicely. So if you have a look down at the bottom here, this here will show you the number for the node. In this case, you can see it starts at 9. Look down the bottom there, that's node 9, and it goes 8, 12, 1, and so on. So now these aren't arranged according to just their numbers 1, 2, 3, 4, and so on. Now you can see it matches quite nicely to the reference genome, but obviously these lines will actually tell you or indicate where things have moved, if you like, or there's differences. So what you can do is if you zoom in to a particular area, so for example here, you can see might be a bit of a difference there where the line disappears at the bottom, and so on. And as you go through, obviously there might be bits of information missing. I think that's the point I'm trying to say here. So for example here, in the above genome, there's quite a large cluster of genes here that are present in the top genome but aren't present in the lower one, in the sort of insertion of something or other at that point. So if you're trying to build a consensus sequence, it's going to be quite tricky from this, because as you look through, so for example here, there's bits where there might be missing information, and you might find that specifically at the contig boundaries. So if you look quite closely at one of these contig boundaries, so for example here, you can see here, there's a bit of difference there. So possibly there's information, like I said, that isn't present. It might be a bit vague, but hopefully that'll make a bit more sense in a minute. So let's just minimize this. You've reordered the contigs, but obviously there's some output files as well. So if you go into the folder where you've saved these alignments, I've called it Mauve, you'll see you've got alignment 1 here and alignment 2. So these are the different number of times it's gone through the alignment process. It's the first time, and it's had another go too. But depending on how different these genomes are, you could have 4, 5, maybe more different alignment processes that have gone on. So if I have a look at this one now, alignment 2, and I open this, there's various files in here. If you ever want to actually go back and open up the alignment, you've got it saved here. In Mauve, you can actually reopen it again. File, open alignment. If you look back in that folder, go to alignment 2 and select this one, alignment 2, it'll reopen that again. So it saves itself automatically. A bit more about the files in there. I know one of the questions that we were asked was, well, this was the first one, identify suitable reference sequence. I suppose, yeah, we've done that to a point. I did it for bacteria, so depending on what you're working with, that's the first thing to do. Reorder the sequence genome. So we've gone about and done that, and obviously, like I said, it was assembled into a number of nodes or contigs, and we've reordered those to match the order of the reference sequence. And then save the new alignment so it can be stored in a FASTA file. In here, you've got a couple of bits of information. As we said, this is the alignment file. There's a few other files here that are related to the alignment as well that Mauve will need. you've got your reference sequence GenBank file. But down here is something called unknown. So this is your unknown genome, and it's called contigs. So if you open this with Notepad, this will give you the order of your contigs here. As we looked through the genome and scanned through it, we decided it started with contig 9, 8, and then it went through like that. So you know the order of the contigs here. But as we said, there might be some sequence information missing at the contig boundaries. This part here, contigs with conflicting order information, what that relates to, have a look at, say, this one here, contig 13. Where you get the conflicting information is where things have moved. So you see there at the bottom, where it says contig 13, that's what we're on at the moment, and as I scan over it, you can see the cursor in the top genome is flicking about from left to right. That means, and the line here is going up at an angle, indicates there's been some kind of movement of some of the genetic material within the genome, between the reference genome and the one that you're looking at. So when you've got in here conflicting order information, that's what it relates to. That, you know, some of these contigs might have parts of their genes in one part of the genome and part in another. Just looking up, some of the contigs might be on the opposite strand, or, well, they might be orientated. There might have been an inversion of a block of genes in relation to the reference genome. So that's what that relates to there. Now, this is the general order of the contigs. Now, I know it's not a FASTA sequence. Just going back and having a look, you have a look in this one here, unknown genome FASTA file. We just open that up. This is the FASTA file. You can see contig 9 here, and then if you just search for the next header arrow and just search down, you can see then you've got contig 8, contig 20, contig 12, and these should all match the order that you've got here. So you start here, contig 9, contig 8, contig 12, and so on. So you have got a FASTA file, but there's one problem with this. It's not a consensus sequence. So you could almost butcher together a single genome file by deleting these. But I think the point is, I've had this before, where when something was arranged in lots of nodes, it was quite often that the breaks in the sequencing, so the node boundaries or the contig boundaries, happened at specific points, and every time you got one, there was a bit of information that was missing. So we actually went back and we used a much longer read sequencing technology just to sequence over these gaps. So there you go. That's pretty much it. So what if you were working with a species that had never been sequenced before? What would you use as a reference sequence? Well, there is a website where you can try, depending on what you're working with, try and find something suitable. Reorder the genome. So we did that. We reordered the contigs using the Mauve Contig Mover tool, and save the alignment so it can be stored in a FASTA file. But it's not a saved alignment. You can't build a consensus sequence using Mauve. It's really just for visualizing. But you can get a FASTA file, but it's just going to be the nodes that have been reordered in there. So anyway, I hope that's helped, and put the video up. Just leave a comment if you need anything else.